Welcome to the CannMed Coffee Talk podcast, where we talk with the leading minds in cannabis science, medicine, cultivation, and safety testing. I am your host, Ben Amirault. I am the Senior Marketing Manager at Medicinal Genomics and proud member of the team that puts on the CannMed Summit. Well, we are getting closer and closer to CannMed 25, which is taking place at the Wyndham Resort in Puerto Rico, June 17th through the 20th. If you are planning to join us in Puerto Rico this June, don't wait to register. Our room block at the resort is filling up fast, and you don't want to miss out on this year's event. If you're still on the fence, I invite you to go to canmedevents.com and check out our speaker lineup, our schedule, and our unique networking events designed to connect you with the leaders who are shaping the industry. We really do have something for everyone at CannMed 25 this year. We have the medical practicum, where doctors and nurses can earn up to 6 continuing education credits. We have the Cannabis Genomics Workshop, featuring more than a dozen of the world's leading experts on plant genomics, breeding, and pathogen resistance. We have a lab compliance workshop covering lab shopping, remediation, and other hot topics. And we also have 20 curated oral presentations from the top researchers in cannabis science, medicine, cultivation, and lab testing, and so much more. And the great part about CannMed is that all of these people from the different areas of the industry come together with the goal to advance cannabis medicine and the industry as a whole. And we really hope that you'll join us this year. And since you are a podcast listener or watcher, we do have a special offer for you. I have a promo code that you can use to get $300 off your ticket package. That promo code is Coffee Talk, all one word. If you put that in the promo code box when you buy your full summit package, you will get $300 off. So don't wait. Pause this episode, use the link in the description to go register, pop in the promo code, get your discount, and get ready to join us at CannMed 25. Our guest today is Dr. Zamir Punja. Zamir is a professor of plant biotechnology at Simon Fraser University in Canada. His research interests include the etiology and management of plant diseases on vegetable and horticultural crops, and the application of plant biotechnology for disease management. Since 2018, his work has shifted to researching cannabis, where his group has described a range of previously unreported pathogens affecting the crop and has evaluated various methods for disease management. Using next-generation sequencing, his team identified diverse fungi, yeasts, and bacteria across multiple plant tissues. Their study reveals that cannabis plants host abundant microbial endophytes, which can impact plant health and product quality. Findings also highlight the influence of growing substrates and biocontrol agents on the cannabis microbiome. During our conversation, we discuss what is the microbiome and how do microbial endophytes affect plant health, identification of common microbes found in cannabis samples, including penicillium and Fusarium, whether meristem tissue culture is a viable microbial sterilization method, the influence of sterilized versus non-sterilized growing media, how growers can use microbiome analysis for early interventions, future research questions on microbial balance and seed microbiome engineering, and more. Before we get to my conversation with Zamir, I'd like to thank this episode's sponsor, Grow Defender. Grow Defender provides long-lasting antimicrobial protection for indoor cultivation and processing spaces. Using patented Gold Shield technology, their EPA-registered water-based products protect surfaces from mold, mildew, bacteria, and fungi for up to 90 days. For more information, visit growdefender.com. Okay, and without any further ado, please enjoy my conversation with Dr. Zamir Punja. Good afternoon, Zamir. Thank you so much for joining us on the podcast.
Hi, thanks, Ben. Thanks for having me on here. Glad to be here.
Of course, it's great to have you back on the podcast. You're one of our favorite guests and favorite CannMed presenters. You always have great information about plant pathogens and plant health. You know, I don't know if I've told you this before, but your CannMed 22 presentation on cannabis trichomes is the most watched video on our YouTube page. It has more
Wow,
than 300,000 views.
that's
So,
incredible. I know when I was getting ready for it, presenting it, I had kind of goosebumps because some of the images that we had presented were just incredible. And so I'm really glad that people are catching on to look at these fascinating structures that obviously contain all the cannabinoids and terpenes and so on. So that's good. That's good news.
yeah, absolutely. It's a visual medium, and those images are amazing. Um, so, CanMed 25, we're happy to have you back. Your presentation this year is going to be about the cannabis microbiome and specifically microbial endophytes and how they affect plant health. But first, for those who may be unfamiliar with those terms, I guess my first question is, can you briefly explain what the microbiome is and how it affects plant health?
Yeah, so I mean, the microbiome was a term that was coined a long time ago for human health, because as you know, we have microbes in our gut and elsewhere, and those microbes interact with various aspects of our body, and some physicians feel that that composition of microbes has various implications for health, not just physical health, but in some cases even your mood swings and various other things. So there's a complex interaction with microbial organisms within human guts, and so that's carried on to plants. I mean, plants, surprisingly or unsurprisingly, also have a collection of microbes that they seem to interact with quite a bit, and those microbes may be in the on the outside.
Mhm.
In the human case, it's all internal microbes, but in plants, it can be a combination of external, primarily things in the soil that get in through the roots. But we also have a subset of the microbiome that's internal, and we refer to that as the endophytes. Endo meaning inside, phyte means plant. So there's microbes inside the plant that, to me, are also pretty interesting because what are they doing there? I mean, are they beneficials? Are they causing issues in the plant? And that's kind of what drove us to start this whole area of research.
Excellent. So yeah, what do we know? Are they what kind of purpose are they serving? Are they helping to carry out certain processes or
Yeah.
what's going on there?
So again, there's a fair amount of literature on these endophytes and microbiomes in other plant species. So things like wheat and corn and tomato. And so we can learn stuff from those plants because again, with cannabis and hemp, I think the earliest studies on microbiomes was around 2020. So
Mhm.
we've only been at it for 4 or 5 years, and certainly for cannabis or the high THC strains, this has only been in progress for about a year and a half. So it's relatively new for these two crops. But in other plants, there's these microbes that sit on the root surface that seem to help absorb nutrients. There's others that fight off, you know, pathogens that might try to gain access to the roots. And then some of these microbes enter into the roots, the beneficials or the endophytes. And again, some of the ideas are that they do have some benefit. Um, Otherwise, why would they be there? So we've taken the opposite approach, is that there may be some in there that are not
Mhm.
beneficial. And some of those would include pathogens, right, that also gain access to the inside of the root structure. And so there's probably a battle that goes on between the good and the bad within the plant structures. That part we haven't quite figured out yet. What we've done up to now, using actually medicinal genomics platform, is to go through and sequence these organisms that are there using, what they call whole genome sequencing, where using your platform, we're able to actually um, determine what the species of these microbes are based on their sequences of DNA. And so we've come up with this quite a large volume of information that we're sort of sorting through. I mean, what does this mean? Is this X? Is this Y? Is this Z? And eventually we'll come up with some sort of answer to explain what these microbes are doing.
Right. And now are you trying to kind of correlate to um, the findings that you have in the microbiome with, symptoms or morphology of the plants themselves to try to make some connections there?
Yeah, that's really hard to do, because what we're finding is that there's no plant, there's no cannabis plant that doesn't have a microbiome.
Yeah.
So you don't have a starting point that's zero. So everything that we take up already has a microbiome in it. So you ask the question, well, if I was to add X and Y, is that going to make a difference or not? But you've already got, A and B sitting in there already. And so it makes the evaluation of these new microbes that you add more complicated.
Hmm.
What's A and B doing there? We don't know yet. And so when you add, X and Y, what does A plus B plus X and Y do together? We're not really 100% sure. There's about two or three recent reports that show that when you do add X and Y back to whatever's there already, there is some benefits that they do see sort of a enhancement of growth. There's also apparently an increase in some of the cannabinoids, but we need to be sure that it's a consistent trend, that when you add X and Y, that you're actually going to see what you think you're going to see.
Right. It must be hard to try to control for all those different variables.
There's a ton of variables, and that doesn't even include the environment or the cannabis strain that you're using or the fertilizer that you might be adding. So that adds a lot of complications to what we might see. And again, similar to the microbiome in our gut, depending on your diet, depending on whether you take in a lot of yogurt, depending on whether you
Hmm.
take antibiotics, that's going to influence your microbiome. And I know yours is different from mine because our diets are completely different. So there's a lot of variables that also apply to plants.
Interesting. Yeah, and when I was looking through your abstract, I did see that there are certain microbes that you were finding in all samples. I believe it was Penicillium and Fusarium. What's the potential implications of that?
Yeah, so that was interesting as well. I mean, the Fusarium we know is a pathogen, so we need to watch out for that one. And I guess in hindsight, it wasn't too surprising because we know Fusarium, once it gets into the mother plants, you take cuttings from those plants, you have a good chance of spreading that Fusarium off onto the next cropping cycle. So we kind of knew that Fusarium was there. And we've also seen a lot of Penicillium. I mean, Penicillium is what the term we use is ubiquitous. It's kind of everywhere. You find it in the air, you find it in the soil. We use it to make cheese. We use it for various things. But we found penicillium consistently, and probably more interesting than that, there was one species, Penicillium chrysogenum,
Hmm.
which in the early literature back in the 50s and 60s, it was described as the species that produces the antibiotic penicillin, which we all know is pretty useful for helping us combat bacterial infections and so on. So what is this penicillin producing penicillium inside the plant doing? Maybe it's fighting off some of the fusarium, maybe it's fighting off something else. But in those two cases, the consistency suggests to me that there's a role, there's a function for that penicillium inside the plant. And I don't know if it's producing antibiotics. Well, that's a harder question to answer, but it's there, and so it must be doing something.
Yeah. And no, and you mentioned that Fusarium is present in all the tissue samples and that it is a pathogen. So are these plants showing symptoms of like a Fusarium infection, or is it potentially at such low levels that it hasn't had a chance to cause harm?
Yeah, I think it's there at such low levels, and that brings up a good point. This microbiome sequencing is very, very sensitive.
Hmm.
I mean, the level of detection may be, a few cells of the species that are present inside the plant. So when we see a positive for Fusarium, it doesn't mean that plant's completely overtaken by it. It means they may be, well, for argument's sake, 10 spores
Hmm.
that we were able to pick up with this analytical method. So it could be just dormant, could be maybe it's being suppressed, by the penicillin that's inside, and then when you allow it to grow further, it explodes. We know a lot of these endophytes are not necessarily expressing something all the time. A lot of them can be what we call latent.
Hmm.
It's an infection that you have kind of like what we used to have when we had COVID with no symptoms. The virus is there, but it hasn't exploded yet. A lot of these endophytes do that. They stay latent. And then if the plant is stressed or there's some other environmental factor, they all of a sudden become active. And that could explain what the Fusarium is doing and possibly the Penicillium.
Yeah, and that was going to be my question because I know that it's been said before that, if you overwater your plants, you have an overly humid environment or it's particularly damp, you're more prone to like a fusarium infection. So is it that, if the conditions are right, it can sort of take off?
Yeah, and it's really hard to get rid of. The only time where we've actually been able to see it completely eliminated, and again, this is using the microbial sequencing platform, is when you do meristem culture.
Mm,
When you take the meristem tip, which obviously is for eradicating viruses and viroids and other things, what we found was that the fusarium was gone. It was gone in those plants that came out of the tissue culture. And then I'll just add something more to that, and I'm sure you're going to have a follow-up question. And that is that when you put those plants out, they get reinfected really fast. So there's fusarium flying around in the environment, and
mm.
for some reason, those meristem-derived plants seem to be, and I haven't proven this yet, more sensitive to being reattacked. So it's almost like you've cleaned your entire gut of the microflora. You've taken a ton of antibiotics,
Right.
and now the danger is that whatever comes in next is going to completely take over because it's created this biological vacuum, right? So they always say when you've done a dose of antibiotics, go back in and start taking these beneficial probiotics
Right.
to help reestablish your gut microflora. It may be the same in these plants. Once you clean it up, you need to make sure you put something useful back, beneficials, which would include things like trichoderma and other beneficial fungi.
Yeah, no, and you're right. I was definitely gonna have a follow-up to that. But before we sort of move on, I just want to talk more about meristem tissue culture, and sort of, the general idea behind that, as I understand it, is you're taking such a small piece of the newest growth that you're potentially outrunning the infection, right? The new growth
Yeah.
is so new that the infection hasn't had a chance to get there yet, which makes a lot of sense. But if I read correctly in your abstract, you were saying that some of the bacteria is still present in the meristem.
Yeah, there were still a few bacterial cells. And, bacteria, first of all, they're so microscopic and they're very, very easy to move through the vascular tissues in the plant. So, unlike the fungi and maybe some of the other viruses that sort of lag behind, some of these bacteria were seemingly making it through.
Hmm.
Is that good or bad? I don't know. It basically means that, the meristems aren't completely sterile. There may be some remnants of some bacterial cells. And we know even with viruses and viroids that you don't get 100% success rate. You may be at 30, 40% elimination. But the fungi, because they're larger organisms, seem to be removed from those meristem tips.
Yeah. No, and I suppose it makes sense too. If the idea is that there's almost a symbiotic relationship between the plant and the microbes, that it would be present even in the new growth because it may very well be
Yeah.
essential.
Yeah, it's doing something that we still haven't quite figured out. Yeah.
Yeah. So another thing that you had touched on earlier was the idea of um, taking cuttings. So if the mother plant is infected with, I think it was Fusarium that we're talking about, that the cutting is going to have those same symptoms. So what are some of the implications there for growers who rely on that?
Yeah, so that's something where you have to be really careful with the Fusarium. But in addition to Fusarium, you're also carrying over some of the endophytes, the good and possibly the other bad ones. What we found was that there was a really high correlation between the overall endophytes that you established using this microbiome method and the endophytes that were originally in the mothers. Which, I mean, in hindsight, you could say, yeah, of course, we know that mother to progeny, you get the transmission, but it also implies that pathogens will move in that
Hmm.
direction as well. And you can't possibly tissue culture everything. I'm not suggesting we go and meristem the entire population of plants, but what you can do early on, at the time of rooting is where we think a lot of these endophytes and other microbiome members are getting established. Is when the roots are starting to be formed. And so we're thinking that as you start rooting, in addition to whatever may already be there, you can supplement that with beneficial microbes. And again, things like Trichoderma, Gliocladium, some of the bacteria come to mind. Add it in early so that as the roots start to form, these microbes can get in there
Mm,
and potentially outcompete the Fusarium and some of the other microbes that are there.
excellent. Yeah, and that was another thing I was going to bring up was this idea of the growing medium. Because I think it was said that sterilizing the cocoa fiber growing medium actually didn't. m- Sorry, you mentioned that sterilizing the cocoa fiber growing medium didn't influence the microbiome of the cuttings grown in them. So, what does that mean in terms of
Yeah. So now
prevention?
that one's a double-edged sword in the sense that there's two aspects to that. The first is that if you've got a mother plant that's already got endophytes or other microbes in it, and you take cuttings off of that, you'll be moving along those particular endophytes. So we call that horizontal transmission. It's sort of moving from one side to the next. Then if you plant that in a substrate, what you'll find is that the whatever's in the substrate, growing medium, doesn't really have a huge impact on whatever has already been established. In other words, there's sort of a buffering
Mm,
effect. So that speaks to perhaps the substrate not having a major effect. However, if you go backwards and consider the mother plant and the mother plant before that, those plants, if they're planted in a substrate that has a lot of microbes in it, they'll start absorbing those microbes from the surrounding environment. So if you look at a time course, time A, the substrate becomes very important
mm.
because the substrate will influence whatever's getting into the plant at time A. But when you take a whole bunch of cuttings all the way to W, X, Y, and Z, those microbes that came from that mother plant seem to be somewhat well established. And then whatever's in the substrate doesn't seem to make a huge amount of difference because they're already established.
Right.
So yes and no, the substrate can have an influence in getting that initial population established. This is where I think the best scenario would be sterilizing that substrate just to be sure, and then adding back in there what you feel are the beneficials, whether they be trichoderma or gliocladiums. Get those set up, and then stick your plants in those. And the idea would be that those will try to outcompete whatever's already present in the cuttings and make it a beneficial environment.
Yeah, I suppose that makes sense. If you keep mom happy, you keep mom healthy, when you're creating your cuttings, try to um, recreate that same substrate for the
Right.
cuttings, and then hopefully that would
And and
continue on.
and I think that's where this whole idea of living soil comes in. I mean, I've never
Mm.
experimented with living soils myself because we work with hydroponic systems where everything has to be, clean. But living soils have the advantage that they also carry a huge complex of microbiomes or microbes that can have some impact on the plant that's subsequently grown in that environment. The only difficulty I have with living soils is that I don't know totally what's in there. I think what we
Yeah.
need to do with the microbiome studies is now start looking at living soils and finding out exactly what it is that we're dealing with. I mean, living soils do differ. If you've added a molasses solution to it or you've added some other organic fertilizer, that's going to influence the microbial composition, and that could then impact the plant. So certainly there needs to be more work in that area.
Excellent. No, I think we've done a good job talking about how endophytes microbiome impacts plant health. What about concerns about from a consumer standpoint?
Yeah, so I mean, one of the because the technique is so sensitive, we're finding a lot of interesting things that you wouldn't have been able to culture or grow on media and so on and so forth. So when you first look at the data, you go, "Oh my God, there's so much stuff here."
Mm,
But we can't lose sight of the fact that every plant, every vegetable, every fruit product that we eat is covered with microbes, right? There's always a microbiome that's associated with the tomato that you eat and the cucumber that you eat. So we can't lose sight of that. But because cannabis is more highly regulated, right? They want to make sure that you don't have the negative microbiomes present. It could help address the issue of these microbes microbes that like Aspergillus that we're really concerned about, how
Yeah.
much of that actually originates with the microbiome itself? And how much of it is actually coming in from the external environment? The data we're seeing suggests that Aspergillus is not huge. It's not huge in the microbiome itself as the plants are growing. So they're not like harboring it and picking it up and storing it like penicillium. It may be that the Aspergillus is an external factor, right? It's coming in maybe through high humidity conditions or poor growing conditions or poor drying conditions. And if that's the case, then it at least allows growers to manage the environment more than they have to be worried about the plant itself.
Mm.
So we're hoping that some of this microbiome eliminates causes for concern, even though you've got such sensitive data that you've got 8 or 10 cells of one microbe, and it may show up as a positive.
Yeah. No, and that's interesting you mentioned Aspergillus because one of the arguments against doing Aspergillus testing is that it's ubiquitous. It's everywhere. You can't possibly be growing without it. So it's interesting to find for you to not find it, I suppose.
Yeah, I mean, at the early stages anyway of plant growth, it may establish itself later on, but it's definitely not one of the high numbers. The ones that I see that we may end up spending more time looking at is Fusarium, because
Mm.
we're seeing
Yeah.
Fusarium early on, all the way through the plant, and in some cases getting into the flowers, and we know that happens, the flower infections. And those species also produce various mycotoxins. And so we know that one can originate from inside the plant. And to me, that may be more concern in the long term than perhaps Aspergillus that could be an external source of inoculum.
Yeah, that is definitely interesting because I know Fusarium is a problem and a concern, and we've developed qPCR testing to try to screen plants for it, which is why it's sort of interesting that if it's so ubiquitous, perhaps there needs to be some sort of cutoff or threshold where it's more of a concern, but that would take
That's
some
going to
more
be that's
study.
the next step. I mean, we don't want to add any more regulations, but I think what we
No.
want to do is make sure that the regulations that we are using make sense, like make biological sense. I know Aspergillus is up there because historically it's had some major issues on human health, but if it's not as prevalent or not embedded in the plant and it's coming from outside and perhaps not as much concern, then maybe we need to reassess what the important microbes are.
Yeah, absolutely. And I know too that you analyze different tissue types from roots to leaves to flowers, stems. What are some of the insights there from looking at the different tissue types?
Yeah, so there's a fair amount of variation there, and similar work has been done with hemp. There's been a couple of papers that have shown that there are tissue-specific groups of microbes. What that means is that you might find one type more prevalent in the roots and another type more common in the leaves and so on. Definitely the roots have the most.
Hmm.
I would say the next tissue that has high levels are stems, which kind of makes sense because that's
Right.
where the transport is occurring. And then less so in the leaves. The leaves seem to have fewer of these endophytes and other microbes. And we're only now in the process of analyzing the flowers, which is the critical phase of the analysis. But we have to be a little careful with flowers because, as I mentioned, there's a lot of external introduction of these microbes from the air, from water, from whatever else. So when we do the microbiome analysis of flowers, it's not showing only what's internalized. They could be external things that come into play for that analysis.
Hmm. So, in terms of giving growers some actionable items from your research, like what I know we kind of touched on some things about using trichoderma or beneficial microbes, but in addition to that, what other sort of advice could you give based on your findings here?
Yeah, so I think if growers have some favorite genetics that they've been hanging on to that they really want to keep going, it would be not a bad idea to test the microbiome of that particular plant or series of plants. You can collect root tissues or you can collect stem tissues and send it off for analysis, and it'll give them an idea of what sort of the background microbes that are already established in the mothers, because we know that a good proportion of that are going to move on to the cuttings and then so on and so forth. So uh, and it may also reveal what other pathogens could be present that they had no idea were there. So um, definitely for favored genetics that are being stored for a long time, go ahead and do a microbiome analysis on it and see what you got. Following that, cuttings made from those plants, I would definitely suggest they be rooted in a medium that has beneficials in it. Can't go into details about which ones. Some growers have preferences over what they consider beneficial. Definitely have those on early. Some growers tend to put it on later when the plants are rooted and they're getting ready to go into flower. I would say put it on much earlier and get those established. Now, one fungal organism that we didn't touch upon is the mycorrhizae. The
Mm.
mycorrhizae being those, we know they're all pretty much beneficial fungi. I was surprised that there is mycorrhizae present already in some of these plants, cannabis plants that seem to have a favorable relationship with the mycorrhizae, and we're finding it in the roots where you'd expect. We're finding it in the stems, which we didn't expect, and we're even finding it higher up in the plant. So it almost suggests these mycorrhizae may be carried along, uh, through the successive development of that plant, which to me was a surprise because we always think of it just in the roots,
Hmm.
like it's just there. And that's definitely a beneficial that needs to be looked at.
Excellent. No, and it's great advice to kind of get your microbiome analyzed. Um, and not to put you too much on the spot here, but if someone were to do that and looked at their report, are there any certain microbes that would sort of be a red flag that they potentially have an issue, and how would they go about remedying something like that?
Yeah, so there's always surprises when you get down to that level of analysis. It's always concerning. Now, one thing that I didn't mention that probably is worth mentioning is that when you do get the microbiome results back, it also reflects cells that may be dead. In
Hmm.
other words, the microbe may have been there, but it's not there anymore. So you're getting a total analysis of the past and the present in terms of what could be there. I mean, we haven't seen any super surprising results vis-a-vis something new and exotic that hasn't been reported before in North America or some other really bizarre. We're finding what we would expect are microbes that grow in substrates, that are microbes that are in the environment. It was really interesting because the one of the hits, we call it a hits, meaning a match. One of the matches we found in these samples was human DNA. And I'm thinking, okay, there's human DNA. Well, it makes sense because people are touching the plants, they're
Sure.
they're watering them, they're doing things to the flowers. So some of our skin cells come off, right? And they land on the tissue. And sometimes they can be mixed up with the DNA that we extract from the plant. So there's some human DNA, which I mean, somebody may freak out and think, "What's that doing there?" It's not surprising.
Hmm.
So it's sensitive enough that some cells from human hair or skin cells could show up in the analysis.
Interesting. So looking forward, I know that you said that we're still very early on in terms of looking at the microbiome for cannabis and hemp. So, what are some questions that you have that you're looking to answer in the coming years?
So for sure, we need to find out what a good balance of microbes is. In other words, you've got to I don't just throw a number out. We have maybe let's say 50 different species that are present in a cannabis plant. We need to narrow that down to maybe the optimal 4 or 5 that we
Mm.
think are always there, that they're serving a good purpose. One being the penicillium, maybe the mycorrhizae, and then see if we can enhance those. In other words, favor the plant so that it's going in a different direction. The interesting thing that there was a report out of Europe just last year. They compared um, cannabis or hemp varieties that had been heavily selected or bred over time versus some that were more or less wild or hadn't been successively bred over the years. And they looked at the microbiome differences between those two. And what they found was that in the ones that were heavily bred, the microbiome variability was way lower than what you'd find in the natural plants or the so-called the land races. So one of the things that's happening as we evolve with more and more sophisticated cannabis or hemp varieties is that we're altering the microbiome
Mm.
composition without even realizing we're doing that. So if we knew what some of these beneficials were, we would definitely want to add those back to that subset population that's being used for breeding purposes to make sure that they have, the necessary background. The other thing I found interesting was that seeds. There's
Mm.
a fair component of the microbiome that ends up in the seed.
Mm.
And then obviously that's carried over to the next generation. And that to me is also pretty fascinating. Can we modify the seed microbiome early on to alter its characteristics so that maybe it begins to show favorable factors. Now, one quick mention, of course, is that we know from the scanning microscopy studies we did where we can look inside the plants themselves. The reason they're in the seed is because we're pretty sure now they're traveling through the vascular system. They're getting into the xylem and finding their way into the seed, if this is a seed plant, and finding their way inside the seed that way.
Wow. Oh, so we're going to see some more great images.
Probably. I haven't looked at the seeds myself yet, but I'll bet you that there's definitely things in there. But I can show you some really nice images of the what we call the plumbing system of the plant, which is the xylem, and you can actually find these spores embedded, all
Wow.
different kinds of spores in there that nobody has ever seen before. It's a visual confirmation that the microbes, the microbiome is shifting, moving up the plant.
Wow. That's really cool. This video might break 1 million when you have that.
Well, let's see. I don't think anything beats the trichomes, man.
Really?
That stuff's the best. The trichomes are the. I mean, this is pretty cool too, but the trichome photos, it still just blows me away.
Awesome. Yeah, and it's good you mentioned seed because I had a question earlier and we kind of had moved on. But when you were talking about how when you take the cuttings from the mother plant and sort of introduce it, the microbiome sort of established. So the substrate microbiome is not going to have as much of an impact. Is the same true for seed? Or when you place a seed into a growing medium, is there more of a chance that it could influence it?
Yeah, that's a good. I would say that a seed that's just starting to grow and putting out a small root would be more influenced
Mm,
by the microbiome. In other words, you can have positive and negative effects at that young, very young stage because it's not as well established as a cutting. The cutting
right.
is, 100 times larger and it's got its own microbiome. But the seed could be influenced by external factors in the substrate. That would definitely make it important.
Excellent, excellent. Okay, Zamir, winding down here, before I let you go, I want to give you a chance to share with the listeners or viewers um, any other resources on this topic that you think might be useful. Be happy to put a link in the show description or just links to any of the work that you're doing or ways that they could potentially get in touch with you.
Yeah, so I mean, we actually just submitted not too long ago that paper that describes the endophytes and other microbiomes in cannabis plants, and it's provisionally being accepted. So it may come out in the next week or two. What I'll do is probably send you a link to that. That's got the latest information on cannabis endophytes,
Perfect.
and maybe that link could be hooked up to the podcast.
Excellent. Yeah, I think people would love to see that. All right, Samir, great stuff as always. Thanks again for joining us on the podcast and look forward to seeing you down in Puerto Rico for CannMed 25.
Thanks a lot, Ben. It's always great to talk to you, and it's nice to be able to cover different topics every time we speak.
Absolutely. All right, we'll be seeing you soon.
Thanks a lot.